Phospholipids and Glycosphingolipids are components of cell membrane. Eicosanoids are extremely potent compounds that elicit a wide range of immune responses. These are complex lipids. Today, we will cover these complex lipids in 3 parts. First, we will start with phospholipids. Phospholipids are polar and ionic compounds composed of an alcohol attached to either diacylglycerol or sphingosine by a phosphodiester bond. Phospholipids are amphipathic in nature. Each has a hydrophilic head and a long hydrophobic tail. Phospholipids are classified into 2 groups. One is glycerol-based. Each contains glycerol. They are called glycerophospholipids or phosphoglycerides. Second is sphingosine-based. Each contains the sphingosine. Sphingosine itself is an amino alcohol. It is the major structural lipid in the membranes of nerve tissues. The polar head of glycerophospholipids contains an alcohol attached to diacylglycerol by a phosphodiester bond. It is polar and ionic. Glycerophospholipids are components of cell membranes in all tissues. They are also a reservoir for intracellular messengers, anchors to cell membrane, and components of bile, lipoprotein particles, and lung surfactant. There are 2 groups of glycerophospholipids. First group is phosphatidic acid-based. Phosphatidic acids are the simplest glycerophospholipids. 2 fatty acids are attached to glycerol through an ester bond. Phosphatidic acid-based phospholipids include PE, PC, PI, PG, and cardiolipin, which will be shown on the next slide. Second group is ether phospholipids. A fatty acid is attached to glycerol at C1 position by an ether instead of ester linkage. 2 examples will be shown. One is plasmalogen, another one is platelet activating factor. Phosphatidic acid-based glycerophospholipids use phosphatidic acid as a backbone. Alcohol is attached to phosphor group at C3 position through an ester bond. When serine is attached to the phosphate group, the result is phosphatidyserine, or PS. Similarly, we will get phosphatidyethanolamine, PE, phosphatidylcholine, PC, and phosphatidylglycerol, or PG. Phosphatidylinositol, or PI, contains stearic acid at C1 position and arachidonic acid at C2 position of the glycerol. Therefore, PI supplies the precursor for prostaglandin synthesis. Both PG and cardiolipin are important components of the inner mitochondrial membrane and bacterial membrane. Cardiolipin is the only human glycerophospholipid that is antigenic. Here are 2 ether phospholipids. First one is plasmalogen. Plasmalogens are abundant in nerve tissue and heart muscle. Fatty acids is attached to glycerol by either instead of ester linkage at C1 position. Synthesis of plasmalogen takes place in the peroxisome. Platelet activating factor is one of the most potent bioactive molecules known today. It is synthesized and released by a variety of cell types. it causes platelets to aggregate and degranulate. Compared with plasmalogen, a platelet activating factor has a saturated alkyl group at C1, acidic group at C2, and choline at C3 positions. Phosphatidic is typically phosphorylated at position 4 and 5 in the inositol ring, leading to phosphatidic 4,5 bisphosphate or PIP2. When the G protein couple receptor is activated, G protein binds phospholipase C and activates it. This enzyme will cleave PIP2, releasing inositol 1,4,5 triphosphate or IP3. IP3 binds a receptor on the ER membrane, causing releasing of calcium from the ER. Some proteins can be tethered to cell membrane. Here is a protein. It is first attached to a sugar chain through phosphoryl ethanolamine, which is then tethered to the cell membrane through phosphatidic. This type of link that tethers a protein to membrane is called glycosylphosphatidic linked or GPI linked. More details can be found in this picture. Here is a sugar chain, and this is phosphatidic. Examples of GPI-linked proteins include alkaline phosphatase, acetylcholine esterase, and decade accelerating factor, DAF. Most of glycerophospholipids are synthesized in smooth ER and transported to Golgi apparatus and then to membranes of different organelles. In most tissues, including the liver, glycerophospholipids can be synthesized either by donation of phosphatidic acid from CDP diacylglycerol to an alcohol or the other way around. Donation of a phosphorylated alcohol from CDP alcohol to diacylglycerol. Nevertheless, some phospholipids have preference. For example, in most tissues, the primary pathways for synthesis of PE and PC involve phosphorylation of ethanolamine or choline, followed by conversion to the activated form CDP ethanolamine or CDP choline. Finally, phosphoryl ethanolamine or phosphoryl choline is transferred from nucleotide to diacylglycerol. The liver, in particular, can synthesize PC de novo even when free choline levels are low. To do so, phosphatidylserine is decarboxylated, giving rise to PE. Decarboxylates require pyridoxal phosphate and activated vitamin B6 as a coenzyme. The next three methylation steps generate PC. SAM is the methyl group donor. The primary pathways of phosphatidylserine is through base exchange with PE. The three cases that involve synthesis of PE, PC, and PS are summarized on this slide. Cardiolipin is synthesized by transfer of phosphatidic acid from CDP diacylglycerol to a pre-existing molecule of phosphatidic glycerol. This reaction is quite unique. It takes place in the mitochondria. Here is the integration with microbiology. Cardiolipin is also an important component of bacterial membrane. Cardiolipin is recognized by antibody raised against Treponema pallidum, the bacteria that cause syphilis. Phosphatidylcholine, also called lecithin, is the major component of lung surfactant. When Pc levels are low, as indicated by low Pc to sphingomyelin ratio, infants have high risk for respiratory distress syndrome. Here are enzymes that cleave glycerophospholipids. Phospholipase A1 cleave glycerophospholipids and release fatty acids from the C1 position. Phospholipase A2 release fatty acids from the C2 position. Phospholipase C cleaves phosphomonoester bond at C3 position and release phosphorylated alcohol. The most renowned example is the release of inositol triphosphate from phosphatidiinositol by phospholipase C. Now let's move to part 1b. Sphingomyelin has a sphingosine backbone. Sphingosine is made up of palmitic acid and serine. Addition of one more fatty acid at the amino group pointed by the arrow, giving rise to ceramide. And this is ceramide. Addition of phosphorylcholine resulting in sphingomyelin. To synthesize sphingomyelin, palmitoyl-CoA and serine first condense to give rise to sphingolin, with removal of CoA and a loss of carbon dioxide. This reaction requires pyridoxal phosphate as a coenzyme. This is very similar to the decarboxylation reaction in the synthesis of PE from PS. Desaturation of sphingolin gives rise to sphingosine. Addition of another fatty acid gives rise to ceramide. Transfer of phosphorylcholine to ceramide gives rise to sphingomyelin. To degrade sphingomyelin, sphingomyelinase removes phosphorylcholine, leaving a free ceramide. Ceramide is then cleaved by ceramidase into sphingosine and free fatty acids. The ceramide and sphingosine regulate cell signaling by control of protein kinase C activity. Now let's move to part 2. Glycosphingolipids are derivatives of ceramides, in which carbohydrate moieties are attached to ceramide. Glycosphingolipids are found in nervous tissues. They play a role in regulation of cellular interaction, growth, and development. They are very antigenic. Here is a structure of glycosphingolipid. This is a ceramide, and this is sugar. Glycosphingolipids do not contain phosphate. Polar head is provided by monosaccharide or oligosaccharide attached to the ceramide through an O-glycosidic bond. Here is a general structure of sphingolipid. Portion shaded in pink is sphingosine. All sphingolipids use sphingosine as backbone. Fatty acid is added to sphingosine at position C2. These together make up a ceramide. Addition of an alcohol or carbohydrate at position C1 gives rise to a sphingolipid. Two conclusions we can make at this point. First, ceramide is a precursor for all the sphingolipids. Second, the X group at position C1 determines the function of the resultant sphingolipid. The simplest neutral glycosphingolipids are the cerebrosides. In cerebrosides, a single monosaccharide is linked to ceramide. Most common cerebroside is galactocerebroside. It is found in neuronal cell membrane. Glucocerebrosides are not normally found in cell membrane. They are intermediates in synthesis or degradation of more complex sphingolipids. Buildup of galactocerebroside is related to Krabbe's disease. Buildup of glucocerebroside is related to Gaucher's disease. Here, a mnemonic Kg may help you put these two diseases together. In most countries, Kg is used as a unit of weight. Here, K represents Krabbe's disease, G represents Gaucher's disease. These are the diseases caused by buildup of cerebrosides. Attaching additional monosaccharides to glucocerebrosides gives rise to globosides. Build up a globoside containing an amino sugar, N-acetyl-galactosamine, is related to Sandhoff's disease. Buildup of a globoside without amino sugar is related to Fabry's disease. Here, a mnemonic SF may help you put these two diseases together. SF is abbreviation for the city, San Francisco. Here, S represents Sandhoff's disease. F represents Fabry's disease. Gangliosides contain acidic sugar nana. In notation, GM2, G stands for ganglioside, M stands for mononana. The number refers to the order of migration of gangliosides on thin layer chromatography. To make it simpler, GM1 is larger in size than GM2, which is larger than GM3, as shown in this picture. GM1 is larger than GM2. GM2 is larger than GM3. Gangliosides are the most complex glycosphingolipids. Buildup of GM1 is related to GM1 gangliosidosis. Buildup of GM2 is related to Tay-Sachs disease. Sulfatides are cerebrosides in which galactoside residue is sulfated. Sulfatides buildup is related to metachromatic leukodystrophy. Similar to phospholipids, synthesis of glycosphingolipids also takes place in the smooth ER and the Golgi apparatus. Similar to glycosaminoglycans and glycoproteins, synthesis of glycosphingolipids occurs by sequential addition of glycoside monomers from sugar nucleotide donors to acceptor molecules. Enzymes involved in synthesis are glycosyl transferase and sulfotransferases. Similar to glycosaminoglycans, TAPS is used as a sulfur donor. Degradation takes place in the lysosome. Degradation occurs in reverse order to synthesis. Last on, First off, here we will start with one enzyme, lysosomal beta-hexosaminidase. It is a dimer comprised of two subunits. Beta-beta dimer makes up a beta-hexosaminidase B. Alpha-beta dimer makes up beta-hexosaminidase A. Both A and B can cleave N-acetyl-galactosamine from globocytes. But only beta-hexosaminidase A can cleave N-acetyl-galactosamine from GM2. Therefore, when alpha subunit is deficient, as in Tay-Sachs disease, GM2 buildup. When beta subunit is deficient, both GM2 and globocytes build up. Again, deficiency of alpha subunit only affects beta-hexosaminidase A. This causes Tay-Sachs disease. Deficiency of beta subunits affect both beta-hexosaminidase A and B. This causes Sandhoff's disease. Enzyme deficiency and the related disease are summarized in this picture. These diseases are caused by buildup of sphingolipids. As a result, they are called Sphingolipidoses. The tissue affected most is the nervous tissue. Typical symptom is neurological deterioration leading to early death, except Gaucher's disease and Fabry's disease. Sphingolipidoses occur more frequently in Ashkenazi Jews than in other populations. Here is Tay-Sachs disease. Mutations in the HEX-A gene cause deficiency of alpha subunit of beta-hexosaminidase A. GM2 builds up, leading to premature death of the nerve cells. Symptoms include progressive neurodegeneration, developmental delay. Very important features include cherry red spot on macula. Here is a cherry red spot. Lysosomes with onion skin appearance. No hepatomegaly. In contrast, Sandhoff's disease is caused by mutation in the HEX-B gene. The mutations cause deficiency of both hexosaminidase A and B, leading to buildup of both GM2 and globocytes. Gaucher's disease is caused by deficiency of lysosomal beta-glucosidase. Glucosylceramide accumulates in macrophages and many other cells. Symptoms include painless hepatomegaly and splenomegaly. Pancytopenia with a low blood cell count. Osteoporosis with abnormal bones. At microscopic level, presence of Gaucher cells. They are lipid-laden macrophages, resembling crumpled tissue paper. Here is the example of Gaucher cell. It is a lipid-laden macrophage. Fabry's disease is an X-linked disease. It is caused by deficiency of alpha-galactosidase A. The deficiency causes buildup of globocytes. Symptoms at early stages include episodic peripheral neuropathy affecting motor, sensory, and autonomic nerves, angiokeratomas with small marks of red to blue color on the skin, as shown in this picture, and and hydrolysis with no sweat. At late stages, patients may develop progressive renal failure and cardiovascular disease. Krabbe's disease is caused by deficiency of beta-galactosidase. The deficiency causes buildup of galactosylceramide, which affects the growth of myelin sheath and causes severe degeneration of motor skills. Symptoms include peripheral neuropathy, developmental delay, and optic atrophy with reduced vision. A key feature of Krabbe's disease is presence of globoid cells that are giant multinucleated phagocytes. Diagnosis can be done using cultured fibroblast cells. Lysosomal enzymes will be tested along with lipids. Here are 3 diseases that have either hepatomegaly, cherry red spot, or both. I use one mnemonic to summarize these 3 diseases: GTP. Here G represents Gaucher's disease that has hepatomegaly. T represents Tay-Sachs disease that has cherry red spot. P represents Niemann-Pick's disease that has both. Now let's move to part 3. Prostaglandins, thromboxanes, leukotrienes all contain 20 carbons. As a result, they are called eicosanoids. PG stands for prostaglandin. TX stands for thromboxane, and LT stands for leukotriene. A third letter, E, F, or I, A, describes the type and arrangement of functional groups. The subscript number indicates the number of double bonds in the compound. Eicosanoids are extremely potent compounds. For example, prostacyclin PGI2 is produced by endothelial cells in response to inflammation. PGI2 causes vasodilation and inhibits platelet aggregation. Leukotrienes are slow-reacting substances of anaphylaxis secreted by mast cells and basophils during allergic reactions. It induces prolonged, slow contraction of smooth muscle and causes asthma. Eicosanoids differ from the true hormones. They are produced in very small amounts in almost all tissues rather than in specialized glands, and they act locally. Dietary precursor of eicosanoids is the essential fatty acids linoleic acid and omega-6 fatty acid. It is desaturated and elongated to arachidonic acid, the immediate precursor of the eicosanoids. Arachidonic acid is incorporated into membrane-bound phospholipids at position C2 of glycerol. It is released from phospholipids by phospholipase A2 in response to signals. After arachidonic acid is released from phospholipids, the first step is oxidative cyclization of free arachidonic acid to generate PGH2. It occurs in 2 steps. The major step is catalyzed by cyclooxygenase, or COX. COX has 2 isozymes, COX-1 and COX-2. COX-1 expressed constitutively in most tissues. COX-2 is inducible in a limited number of tissues in response to inflammation. Further modification leads to different prostaglandins and thromboxanes. The first step in synthesis of leukotriene is oxidation of arachidonic acid, catalyzed by 5-lipoxygenase, which generate a hydroperoxy group at the C5 position. This compound is reduced spontaneously. The same enzyme acts on the reduced compound and converted it into leukotriene A4, an unstable epoxide with this group. A second enzyme, leukotriene C4 synthase, converts leukotriene A4 to C4 by conjugation with glutathione. This is glutathione. Removal of glutamate from the glutathione portion of leukotriene C4 gives rise to leukotriene D4. Further removal of glycine yields leukotriene E4. Leukotriene C4, D4, and E4 are components of slow-reacting substance of anaphylaxis, chemicals released by mast cells during allergy. They are mediators of allergic response and inflammation. Their synthesis is not affected by NSAIDs. Here is a summary of these eicosanoids. Significant ones are prostaglandin E2 and F2. They are synthesized in most tissues. They relax the cervix and cause uterine contraction. Therefore, they are used to induce labor. PGI2 is synthesized in endothelial cells. It causes vasodilation, but it also inhibits platelet aggregation. Thromboxane is synthesized by platelets, opposite to prostacyclin. It promotes platelet aggregation. In addition, it causes contraction of smooth muscle. Leukotriene C4, D4, and E4 are synthesized in mast cells and basophils. They promote contraction of smooth muscle and bronchoconstriction, leading to asthma. Cortisol inhibits phospholipase A2 and blocks arachidonic acid production. Aspirin, indomethacin, and phenylbutozones are non-steroid anti-inflammatory drugs, or NSAIDs, that inhibit both COX-1 and COX-2. Aspirin irreversibly inhibits COX-1 and COX-2 by acetylation at the serine residues of this enzyme, as shown in this picture. Here is a summary of inhibitors of eicosanoid synthesis. When COX-1 and COX-2 are inhibited by NSAIDs, more arachidonic acid is directed to leukotriene pathway, which causes asthma in some patients who use NSAIDs. Thromboxane A2 is produced by COX-1 in activated platelets. It promotes adhesion and aggregation of platelets and contraction of vascular smooth muscle. It promotes formation of blood clots. Prostacyclin PGI2, produced by COX-2 in vascular endothelial cells, inhibits platelet aggregation and impedes thrombogenesis. Although synthesis of both thromboxane A2 and prostacyclin is blocked by aspirin, the inhibition of COX-1 by aspirin cannot be overcome in platelets which don't have nuclei. Therefore, overdose of aspirin induces bleeding.